Welcome. I'm delighted to help host today's ICANX talk and the subsequent discussion, and you're in for a real treat today. Let's go on to the next slide. We have Professor Pupa Gilbert, and then a panel that will introduce after the talk. Professor Gilbert is going to tell you about her extraordinary and exciting science. I'll apologize in advance that I'll actually have to leave during the discussion to go catch an airplane, but I wanted to be here for the talk and the beginning of the discussion. I'm Paul Weiss. I'm at UCLA and the editor of ACS Nano. So for today's talk, we have Professor Pupa Gilbert from the University of Wisconsin at Madison. You'll see her extraordinary look at nature and biomineralization from a number of different viewpoints that really informs how we look at these processes. She's the leading figure in this field and actually fits very nicely with the talks that we had in our ACS Nano lectures earlier in the week. She's originally from Italy, where she was knighted. She's a distinguished professor at the University of Wisconsin at Madison. She's won awards for science, for teaching. She was the head of biological physics at the American Physical Society, wrote a book on physics and the arts, which is what she also sometimes teaches, which has been translated into Chinese. And I know much of our audience is from China, so you can find her work there. And I want to leave her enough time to tell us about her very beautiful work. And it's my very great pleasure to introduce my friend and colleague, Professor Pupa Gilbert. The floor is yours.
Thank you very much, Paul, for that lovely introduction. And thank you to Heixia Zhang for inviting me to give this talk, which honors me greatly. And that's a photo of her. She's a superhero organizing all of these talks. She's an amazingly beautiful, creative, and busy woman. And here she's portraying her Wonder Lab at Peking University. I am standing on the shoulders of giants, literally, not just Paul, whom you just met as the moderator, but also Mikhail, Murong, and Ling, who are going to be in the panel today. So I am literally standing on their shoulders. Look, this is Paul's shoulder, Mikhail's shoulder, Murong's shoulder, and even Ling, who's trying to get away from under me. He's actually under me. That part of his shoulder is under me. He's supporting my weight. So thank you for supporting my weight, for letting me stand on your shoulders today. So today I'm going to talk about biominerals. And in particular, assuming that you guys don't know anything about biominerals, about what mesostructure means, I'm going to tell you about the biomineral mesostructure. I'm also going to assume that you don't know what convergent evolution is, so I'm going to define it from scratch. And then I'll demonstrate to you that some aspects of the biomineral mesostructure, by no means all, but some of them, underwent convergent evolution. And these are in particular amorphous particle attachment and the small misorientation of adjacent crystals. So why should you care about this? Why is it an exciting field? Especially if you're young and you are the creative type, I strongly recommend you join the field of biomineralization. It's one of the most exciting fields you can possibly think of for multiple reasons, but most importantly is because there's room for young people that are creative to actually make big, fundamental, important discoveries. And a lot has not been discovered. So I know it's crowded out there. I know there are millions of scientists. And when you're young, you're very intimidated by the scientific community and you don't know how to find your way. However, there are already skills that you've developed. whether you are a mathematician, a physicist, you're really good at modeling, you're really good at experiments, you're a material scientist, you're an engineer, you're good at mechanics, you're good at optics, you're good at microscopy, you're good at spectroscopy. Doesn't matter what you're good at. Apply that skill that you already have today at your disposal and explore a biomineral system. I promise it will reward you. You'll discover new things that had never been observed before, and they will be fundamental, important, and they will lead to a very high profile publication. How do I know that? Why do I have such confidence that I can promise this to you, not even knowing who you are and what your skill is? It's because pretty much any biomineral that has been looked at led to fundamental discoveries, and fantastic new things that are important have been published very well. And very few organisms have been thus far studied and explored and understood. And yet we've learned quite a bit about biomineralization in general. And we have actually only started to scratch the surface, because the majority of other organisms have not yet been explored, which means that they remain to be discovered, and a lot of fundamental properties are still to be explored. So if you're a material scientist, for example, you can observe very cool structure performances, such a property relations. And sometimes you start from the structure, sometimes you start from the properties. Doesn't matter. You can find those amazing correlations. If you're a biologist, you may be interested in the fundamentals of animal life on Earth and the evolution and how life has changed over the course of the last 500 million years. And biomineralization has very much enabled that. So what are biominerals? Let me give you a very brief introduction to them. You're obviously familiar with your own biominerals, your teeth, your bones. Think about it from an animal point of view. Pretty much any animal needs a rigid skeleton to be able to walk, swim, or fly. Any kind of locomotion needs a hard skeleton. But beyond that, and the major focus of today's talk is going to be on calcium carbonate marine biominerals. And calcium carbonate occurs in 3 different polymorphs, aragonite, calcite, and butterite. All 3 have the same chemical formula, CaCO3. And in this case, what you're looking at is the aragonite coral skeleton that provides shelter to entire coral reef ecosystems. So not only do the tiny little polyps on each one of these tiny little bumps on the surface of this coral skeleton build a home for themselves, or a skyscraper, really. It's not a single home, it's very many homes. There's a whole colony of these genetically identical polyps, but they actually provide shelter for much more than themselves. A lot of little fish, a lot of crustaceans, a lot of mollusks, a lot of bacteria, a whole ecosystem of coral reef depends on this scaffolding, this 3-dimensional infrastructure that coral skeletons made of aragonite provide. That's a photo that I took earlier this year in French Polynesia. These are other photos that I took a little while ago in Bolinas, California, that show that sea urchins that make their biominerals tests, the spines, the teeth, the spicules in the embryos, all of them are made of single crystals of calcite. So that's the other polymorph of calcium carbonate. And the way they use these teeth, these are my hands holding this one sea urchin upside down. You see that at the bottom of the sea urchin, normally what's typically towards the rock, are these 5 teeth that move like the fingers on my hand, open and close. And they are strong enough to actually chew rock. If you look at these holes that they carved into the rock, they carved them just the right size so that they fit into the hole and they find shelter there. They may leave when they go for preying on other organisms and feed themselves, but then they get back home. They're basically condos. And as the sea urchin grows, the hole grows because they keep carving it. They also self-sharpen. The tip of the tooth is constantly sharp the entire time they carve rock. So that's another super fascinating characteristic. They're made of calcite. The substrate rock is limestone that's also mostly made of calcite, and yet they're able to carve it. And that's because they're continuously forming. Biominerals are obviously the most durable part of any animal. Compared to all other tissues, they are the hardest, the stiffest, the strongest, the toughest, and so on. So they actually last a lot longer, not only during the life of the animal, but also after the death of the animal. They're much more likely to be preserved in the fossil record. And in fact, most of what we know about the history of life on Earth was first discovered and put together, making a tree of life based on the morphology of the fossil record. And then, of course, there have been great refinements with chemistry and isotopes first, and then with genome analysis. But the very first evidence was from the fossil record. And even all of those molecular clock estimates are then registered, that they're actually made to correspond to the fossil record. That's an image that I took a few years ago in Lyme Regis. It's a place in the south coast of England that's called the Jurassic Coast. And everything you see there is 200 million year old, and there are fossils on the beach. It's unbelievable. And the beach is eroded all the time, so you see new fossils all the time. It's incredibly beautiful. So here I'm showing you diverse macrostructures that are all made by marine organisms, and they're all made of calcium carbonate, calcite, or aragonite. So here on the top left, you can see echinoderms. So these are sea urchin tests. This sea urchin still has all of its spines. So these spines here have been separated by their test because I bleached that particular gigantic sea urchin. There was this one, many other smaller ones. Then you see mollusk shells here. Some of them had this beautiful iridescent nacre. Some of them don't. And some of them are gastropods, cephalopods, and bivalves. And here on the right, you see corals with this beautiful morphology changes from one genus and species to another. They are dramatically different. Some of them are table-like, some of them are branching, some of them are massive, some of them are encrusting. So you name it, there are 5 different morphologies of coral skeletons. They're all always made of aragonite. The echinoderms, including the sea urchins and the sea stars, are all made of only calcite, and the mollusks are made of both, most frequently. And if they had to choose one, usually they choose aragonite. Interestingly, despite that incredible diversity at the macro scale of mollusk shells, let's focus on the nacre-forming mollusk shells only. And these are the gastropod. This is a California red abalone at the bottom, the biggest one of the shells. A bivalve, this is the pinctada pearl oyster that makes these beautiful pearls that are the Tahitian pearls. And then a cephalopod, Nautilus pompilius from the Philippines. All 3 of them make nacre, and at the micro scale, nacre is actually morphologically identical. This is why it looks iridescent, and this is why it's shining. It's basically a one-dimensional Bragg reflector, if you know a bit of optics. And if we look at nacre, we see that it's actually similar across these 3 completely distinct classes of mollusks that did not learn how to make nacre from one another. They did not have an ancestor that was making nacre. They basically reinvented it from scratch and make it so-called convergently. So what do I mean precisely by nacre? It's one of the most famous biominerals, but in case you haven't heard about nacre, let me show you the mesostructure of nacre in these diverse mollusks. And by mesostructure, I mean the mesoscale, which is from 10 nanometers to 10 micron. And by structure, I mean the size, shape, orientation, arrangement of crystals at that scale. So if you look at the top view of nacre, whether it's in an abalone, a nautilus, or an oyster, you see that it looks like a pavement of irregular polygons. It's like tiles on a floor. And mathematically, this is what's called the Voronoi construction. There is a certain probability of asynchronous nucleation, therefore, of each one of those nacre tablets. Therefore, the sides of the polygons are not perfectly straight. Some of them are a little bit curved. You see, this is a little bit curved here, and this is a little bit curved in the mathematical construction, Voronoi construction. But by and large, these are polygons. If you look from the side instead, nacre looks like a brick wall, just like one brick, another brick, and another brick separated by organic sheets. And the organic sheets are formed first, and then by the cells that are on top in both kinds of nacre, and then the mineral grows to fill this spaces between the sheets. So it's like the floors in a building, and from the staircase comes all of this mineral that invades all of the three-dimensional volume at every floor. So here you have at the bottom in columnar nacre, which is the type of nacre formed by nautilus and by abalone, so by cephalopods and gastropods. Columnar nacre has columns or co-oriented tablets. These ones at the bottom have already fully grown. They've already abutted one another. They've already filled space. The ones at the top have not yet filled space. So this one is growing to the right. This one is growing to the left, and they'll meet each other in the middle. These ones will meet a little later. These other ones later. This is why these look like cones, basically. Same story here in sheet nacre, but they're formed from a side. And so, again, one tablet at a time is being formed. If we look at them with a method that I introduced that's called peak mapping, we recognize that columnar nacre, that sheet nacre. And so I forget to say that in sheet nacre, the columns are actually staggered diagonally, as you can see here. And so the co-oriented tablets are staggered diagonally. If we look with this method I developed, that's called polarization dependent imaging contrast mapping, you can see in different colors different orientations of the crystalline z-axis. Up until this work, everybody said that nacre had in the z-axis of each aragonite tablet, there was a single crystalline, it still is a single crystalline, the z-axis oriented perpendicular to the nacre layers. And that's just not true, because if that was true, here we have in these peak maps different colors corresponding to different orientations. If it was true, we would see one color. If the z-axis was vertical and perpendicular to the horizontal layers in all images, all you would see is cyan. But instead, you see bluish cyan to greenish cyan. And you see that immediately adjacent tablets go from cyan to bluish cyan, which in this color bar actually means plus or minus 15 degrees from the normal to the nacre layers. So a 0 degree C-axis would be perfectly perpendicular to the nacre layers, and it would be cyan, slightly oriented towards, let's say, not quite 1 o'clock, but it would be at 12:30. It would be halfway between noon and 1 o'clock. This would be plus 15 degrees, would be greenish cyan. At 11:30, it would be bluish cyan, and these are all the colors that you see. So you see that they're slightly different. It's not very wrong that all of the C-axes are vertical. They're close to vertical, but they can actually be within a cone of aperture 30 degrees. So interestingly, not only do we see stacks of perfectly co-oriented tablets, or almost perfectly co-oriented tablets, you see these ones are blue, cyan, blue, cyan, blue, cyan, or staggered diagonally in sheet nacre, but we can also see in fossil nacre precisely the same colors, precisely the same structure in a 13 million year old shell. And actually going back into the Ordovician, we can see the earliest formed nacre was 450 million years ago. They all had precisely the same structure. The only thing that changes, and you can see the iridescence because it's a layered material, there's interference with light, there's iridescence exactly the same way in the modern and the ancient. The only thing that changes a little bit is that some of the organic layers that are between nacre tablets actually decay with time in the fossil record. And so as you go back in time, you find fewer and fewer of these. Michael Katz and his brilliant graduate student Jad Salman developed this fantastic method, hyperspectral interference tomography, to actually automatically measure the tablet thickness in nacre and do so non-invasively. So without destroying the shell, which for PIC mapping we very much need to do, we need to actually cut a piece of shell, embed it into epoxy, polish the surface, coat it with 1 nanometer of platinum, and then image it at the synchrotron. We don't need to do that with the hyperspectral interference tomography. You can just look at the entire shell, in this case, Reda Baloni from California and Mikhail and Jad are perfectly able to actually measure the tablet thickness and display it in color, as you can see here, and they find perfectly consistent results with the ones measured with either scanning electron microscopy in a cross-section or what we measure with PIC mapping. And I'm not going to give you any details about this. Suffice it to say, it was published earlier this year in a PNAS paper, and it's been a thrill to collaborate with Mikhail. So I mentioned convergent evolution of nacre. What exactly do I mean by convergence? So if we look at the tree of life, we find these nodes, which means that previously these organisms were all together, and then 686 million years ago, they split. So in particular, mollusks and echinoderms diverge from one another, diverge from cnidarians. Cnidarians are the ones, is the phylum that includes not only corals, but also anemones that are basically corals that don't make a skeleton, and jellyfish. So they diverged from the cnidarians 686 million years ago. And this was estimated with the molecular clock. And there's a big error bar there, like plus or minus 20 million years. But it doesn't matter because we're looking at giant differences here. Mollusks and echinoderms instead diverged from one another 642 plus or minus 20 billion years ago. However, biomineralization started much later. If we look at the oldest fossil age that we know about, for cnidarians, it's 510 million year old. For echinoderms, 520, for mollusks, 535 million years ago. And if we put all of these numbers on an axis of time, you see that there's a list considering those large error bars. In the most conservative estimate, 50 million years between diverging from one another and starting to make a biomineral. What that means is that these organisms evolved biomineralization convergently. This is what it means. They didn't have a common ancestor that already made biominerals. They actually reinvented or reintroduced biomineralization from scratch. So translate mentally, whenever you hear convergence, think independence. Okay, they reinvented it on their own, not inherited. So biomineralization itself is convergent because making biominerals out of calcium carbonate is a convergent aspect. Other convergent mechanisms, I'm going to be talking about two in particular today. One big one is that crystallization by amorphous particle attachment appears to be convergent because we observe it again and again. And the evolutionary advantage that it provides the organism, we think, is that it makes it possible for organisms to grow crystals of aragonite, calcite, or vaterite faster. So achieving faster crystal growth is possible with amorphous precursors. So if you look at all of the mechanisms that lead to crystal growth, conventional crystal growth of anything, biological or not, you think about the bulk single crystal, and that usually grows from solution or from a melt by monomer attachment. So those monomers could be ions, atoms, molecules, doesn't really matter. One at a time gets attached and the crystal keeps growing. This is conventional or classical crystal growth. There's also non-classical crystal growth, and all of these mechanisms that have intermediaries are reviewed in this paper in Science that's collecting an insane number of citations. It's like more than 1,000 now. So these intermediate precursors to crystallization could be crystalline, they could be amorphous, they could be solid, they could be liquid, they could be complexes or oligomers. There's quite an assortment of them, and then they have other intermediaries. But the important part, because this happens geologically, happens biogenically, happens synthetically, all sorts of different things. I'm going to focus on one only right now, the convergent one, which is the amorphous nanoparticles that are formed by the organism, aggregate to form a space-filling amorphous solid, and then crystallinity propagates within that amorphous space-filling solid. So this is the evidence that we have for it. We are looking at coral skeletons made of aragonite, at abalone nacre, also made of aragonite, and sea urchin spines or spicules that are made of calcite. Despite the fact that these end up being either aragonite or calcite, we find the same amorphous calcium carbonate precursor faces to them. We can look at them spectroscopically. You see at the bottom this blue spectrum for aragonite, and for calcite are completely different. Whereas the two intermediate precursors, hydrated and anhydrous amorphous calcium carbonate, which I abbreviated ACC here, is in red or green, and these spectra are identical to each other, whether the final product is aragonite or calcite. If we do a spatial distribution of these spectra with 20 nanometer resolution using a method that I also introduced that's called the component mapping, we see that the most amorphous particles are always outside the skeleton, and at the forming surface of the skeleton, we see quite a few amorphous pixels. Lots of them are red or cyan or magenta here, and magenta means red plus blue, so part of it is still hydrated ACC, part of it is blue. aragonite, and that's what these magenta pixels mean. Whereas in the mature part of the skeleton, away from the surface, this is fully formed crystalline aragonite, totally solid blue. If we look at nacre, here the tablets have already fully filled the space. They are no longer forming, and you see that it's mostly blue already. Whereas in these cones of still forming tablets, there are lots of amorphous pixels. Here you're looking at a sea urchin spicule from a sea urchin embryo, and that's a triradiate one. These are two cylindrical spicules that are perpendicular to the image plane. And you see calcite crystals in blue at the center of the spicule in both orientations of this polished cross section. And the amorphous pixels, mostly green, some red, some magenta, at the outer edges, which are the most recently deposited ones. If you look with the scanning electron microscope, you see that all three of these biominerals, especially after cryofracturing, appear nanoparticulate. What I mean by cryofracturing is that we drop the biomineral in liquid nitrogen, break it with a pair of pliers in liquid nitrogen, so it becomes extremely brittle when it's very cold and it doesn't smear. We can totally see that it breaks like cement would. So it's formed by an aggregate of nanoparticles, and then space is filled by ion growth. So you see that at the same scale, the scale bars at the bottom here are exactly 200 nanometers for all of them, and they're precisely identical. And that's for a coral skeleton, for a balloon nacre, and for a sea urchin spine. And instead, comparing it with a geologic single crystal of aragonite, also cryofractured, it's completely featureless at precisely the same scale. If we look at calcite, it cleaves perfectly, so it's even more atomically flat, even more featureless. What I want to focus on most of the time today is actually the convergent mechanism 2, which is slight misorientation of adjacent crystals. And the evolutionary advantage in this case is greater toughness. So what do I mean? Let's start from a simplest case. This is a spherulite. It's actually not a biomineral one. It's a synthetic aragonite spherulite that was grown in my lab in medicine. And aragonite, again, is a polymorphous calcium carbonate. And what we observe is that in this spherulite, we have acicular crystals. It means needle-shaped crystals that, just like the radii of a sphere, start at the center and radiate away from it. So they look like the radii of a sphere. And obviously, they're only slightly misoriented with respect to their neighboring crystal, typically within 30 degrees, just like nacre. Remember? 30-degree aperture cone. And here we find exactly the same thing. There are tiny little lots of crystals. We measure quantitatively what the angle of misorientation is or two adjacent crystals, and we find that it's usually within 30 degrees. If we look at the coral skeleton of aragonite, this was aragonite, this is aragonite, and that's a coral skeleton. We take one of those branches, embed it and polish it, and then we image it again with PIC mapping, and we see this redistribution of needle-like crystals indicated by the arrows, but you can see a lot more of them radiating like a spherulite, but also when the crystals come pointing at you, perpendicular to the image plane, you can see that the orientation represented by color according to this color bar varies gradually from red to magenta to blue, red to magenta to blue, red, magenta, blue, red, magenta, blue. Fascinating that they all go precisely in the same direction. Isn't it crazy? This is a mad jumble of spherulites in three-dimensional space, and yet there are some regularities which we have not yet fully understood. So, if we look at adjacent crystals within one of those spherulitic bundles of needle-like crystals, we define regions of interest, measure the orientation of one of those corals, and then measure the orientation of the adjacent one using a code that we've actually written, but it's very similar. If you're familiar with Adobe Photoshop, it's similar to the Magic Wand tool in Photoshop. So basically, we do the same thing. Select one region that's defined by a color that quantitatively means crystal orientation in a thick map, and then measure its orientation, the orientation of its z-axis in three-dimensional space. So we do the dot product of those two unit vectors in three-dimensional space and find the angular distance. So if we plot all of the angular distance for a lot of different coral crystals that we've measured, we find that the distribution is very narrow. The maximum misorientation is usually, or maximum angular distance in dimensions, is usually within 30-35 degrees. And we see that the maximum is never a zero and it's never random. It's always a narrow distribution with a small number, maximum like 5 or 10 or 15 degrees, something like that. If we look at synthetic sterolites, there's absolutely nothing biological, nothing controlled, nothing magical here. It's very simple chemicals added to a solution, and as sterolite grows, it does it by itself. We also find a narrow distribution of z-axis. If we look at instead at a completely random orientation of crystals that's in sprinkles, we see that they're all over the place on the horizontal axis that for all of these graphs goes from 0 to 90 degrees. These are all over the place. Whereas if we compare three completely different morphologies of corals from totally different coral genera and species, we find that all of them had this narrow distribution of angular distances, and all of them below 30-35 degrees. If we look at vaterite, that's the other polymorph of calcium carbonate. And this is a tunicate for Herdmania momus, called Herdmania momus. This is a sea squirt, basically, that makes these tiny little spicules to make its body extremely elastic and resistant to the movement of the waves. We find that the adjacent crystals are also narrowly distributed, narrowly misoriented with respect to one another. And this is measured quantitatively and accurately by coding it by my brilliant graduate student, Kayla Stifler. And we find that you've learned the story, that this is a narrow distribution, all of them below 30 degrees, with a few exceptions. There are a few outliers, but by and large, it's a small misorientation angle of adjacent crystals. If we look at calcite on the outside of a pearl oyster, Pinctada margaritifera, we find again a small misorientation. So we are looking at very similar colors. And when there's an abrupt change, like here we go from black to yellow, I should have said earlier, actually, these thick maps are displaying colors. So there are two coordinates. In polar coordinates, you have two angles to define the orientation of the z-axis of any vector. And it is an in-plane angle and an off-plane angle. So the in-plane angle, and that's in the polarization plane of the X-rays that are illuminating the sample surface, the in-plane angle is represented by hue. And so we go from -90, there should be a minus there, it's being covered by another image. So -90 to +90, and all of these hues in between, so red, magenta, blue, and so on. And on the vertical scale instead, this is the off-plane. So when the beam is pointing when the z-axis is pointing directly into the beam, there's no polarization dependence, and that's totally black. So as the crystal rotates, absolutely nothing happens from the polarization point of view, and so peak mapping displays black. And when it's in plane, full brightness color, and you are at the bottom of the scale bar, and every brightness level in between. So every color from green to black, from yellow to black, to red to black, blue to black, and so on. Every color varies in brightness according to this off-plane angle. So I was saying, even when the angles change a lot, like from yellow to this very dark blue here, that's a huge angle. It's more than 90-degree variation. Then you always find this black layer in between. These black layers here are organic. They show no polarization dependence. This is another organic layer. That's another organic layer, which are very well known to exist in the prismatic layer of pearl oysters and any other shell, in fact. Wherever the misorientation is large, then the organism puts a thick organic layer. This happened identically in teeth. We find very small misorientations, and whenever the misorientation is large, there's an organic layer. So this is a shortcut that organisms are using to make organic materials, which are for them a lot more expensive energetically to make, only where strictly needed because the misorientation is large. Now you know the story. The misorientation of these crystals is mostly small, but there are a few outliers, as you can see here, and that's where the organism is putting the organic layers. So peak mapping is by no means the only method you can use to look at these orientations. If you accept a much larger scale, you can actually measure it with EBSD. So have you noticed that I had approximately 5 micron scale bars? Here we have 200 micron or 100 micron scale bars. So it's much lower magnification, but you can use electron backscatter diffraction in a scanning electron microscope. It's a much more conventional method, but extremely useful to measure orientation of crystals. And so this isn't work from my group. This is actually from the group of Igor Zlotnikov at TU Dresden in Germany. And he demonstrates that in Pinna nobilis, each prism in the prismatic layer on the outside of the shell here is a single crystal of calcite. So each one has a single color. And you can see in what's called the pole figure that there's diffraction in one direction only. And so for each one of those crystals, you have one orientation. For each one of these prisms, you have one orientation. It's a single crystal of calcite. For Pinctada nigra, instead, you see these gradients. He actually forgot to put the color bar in these images, so I don't know quantitatively what these gradients mean, but I'm pretty sure they mean the same things that these ones mean at the micron or nanoscale, because they are gradients of color. Their colors vary gradually. So we can safely say, even though I don't know what colors quantitatively mean, we can safely say that they vary gradually, and that's clear and known to happen in Pinctada shells, in all of the pearl oysters. So I told you the evolutionary advantage hypothesis is that this small misorientation that we seem to be observing in a lot of different, completely different organisms demonstrate this. And yes, we did that in 2 different ways. One is molecular dynamic simulations in collaboration with Marcus Buehler and his brilliant graduate student, Andrew Liu, who did molecular dynamic simulations of 2 crystals. So what you're looking at here are crystals at the bottom that had their z-axis horizontal and in the image plane. The top crystals instead have either horizontal and in the image plane z-axis, or slightly tilted. This one goes down 10 degrees and this one goes down 45 degrees. The z-axis, I mean, goes down 45 or 10 degrees or 0. So these 2 crystals are parallel to each other. All of this are bicrystals, and they're actually centered at high pressure at 1 gigapascal in the molecular dynamic simulation. So they become fused together. centered. These two were same orientation, so they became a single crystal. These ones did not become a single crystal. They're still two separate crystals with a well-defined interface between them with lots of defects there, but an ideal system to test what a crack does at crystal orientation interfaces. So Andrew then placed a notch at the bottom of all of these bicrystal systems so that then he can apply a tensile load horizontally, so separate left from right of each one of these bicrystal systems, and a crack will start opening up from the bottom and propagate through the interface, moving up in these images. So let's see what happens when he actually does this. Beautiful. He sees that the crack goes straight, in this case in Aragonite, when the two bicrystals are basically a single crystal. They are centered together and they have precisely the same orientation. The crack is very much deflected at the interface when the misorientation is small at 10 degrees and a lot less deflected at 45 degrees, a greater misorientation angle. That's one of the strengths of molecular dynamic simulations, is that you can also, besides looking at the pretty images we just saw in the movies, you can also quantitatively measure toughness by measuring the area under the stress-strain curve, and you do so from 0 to total fracture. And so this integral that's actually highlighted in this blue shaded area is measured quantitatively, and that represents the toughness. So we have stress, strain, and the integral between 0 and total fracture. So what happens is that's the system I just described to you, C-axis horizontal, slight misorientation or big misorientation, and Andrew did this J integrals and this molecular dynamics simulations for all angles between 0 and 90 degrees for all materials, aragonite, calcite, butterite, and look at these fantastic results for the J integrals. I am super excited about this. Peaks as small misorientation angles for all of them. For aragonite, we see that there is a maximum at 10 degree misorientation of adjacent crystals. For vaterite, there is a maximum at 10-20 degrees. For calcite, there is a maximum at 20-30 degrees. But for all of them, small misorientations, which corresponds perfectly with what we observe with peak mapping at the nanoscale in our synchrotron images in all of the animals. And these are perfect bicrystals. They have absolutely nothing organic, absolutely nothing biological in them. They're just pure atomic systems. Thus, a material property, in this case toughness, may have guided selection and therefore evolution of a structure, a characteristic of the biominerals, in this case, slight misorientation. And I'd like to point out that the slight misorientation toughening is a nanometer scale property, as I showed in the peak maps and in the molecular dynamic simulations. But if this is confirmed, and I'll show you experiments to confirm it in a moment, but this would explain why it evolved convergently. And this convergent evolution is actually a macroscopic scale property that we observe. To do the experiment, to confirm the slight misorientation toughness, we actually teamed up with Davide Ruffoni at the University of Liège in Belgium, and his brilliant graduate students, Alexandra Titz and Astrid Cantanessa. And this is one of our recent meetings. We meet very frequently. This is my graduate student, Connor Smith, who grows the synthetic spherulites. This is Kayla Stifler, who does all of these misorientation measurements. Davide Ruffoni, who's an expert in material science and nanoindentation, and his great graduate student, Alex. So what we did to confirm this slight misorientation toughening was the measurement or rather what we would like to do is to measure Kc, which is the fracture toughness. That's a numeric parameter, and then the Young's modulus divided by the hardness of the material to the 1/2 power, multiplied by the load that's applied by the indenter, and we use a cube corner as an indenter, and then measure the length of the cracks that propagate from the corners of that hole that the indenter makes in the material. And C is the crack length measured from the very center of the hole, of the tip, if you wish, and the tip of the crack. So this we have not yet measured. The experiments are undergoing right now. We had thousands of indents. They're actually taking time, and they're going to have to be analyzed very carefully. So we didn't make it in time for this talk. But what we can do is to actually plot E over H and show you the square root of E over H on the vertical axis here for each one of the systems. So the fifth system that we analyzed is Aragonite, and we saw that the C-axis that's oriented along the long axis of all of these clusters, these are it's a geologic cluster of crystals of Aragonite. The C-axis is always oriented along the axis of these hexagonal prisms that are very clear. Even, you hold it in your hand, it's macroscopic, it's hexagonal, and it's straight, has straight edges, and it has the C-axis perfectly understandable along the axis of the prism. So that's how it's oriented. So what we report here is with the C-axis horizontal, meaning I embedded this sample into epoxy so that the C-axis is in plane and then polished through. Obviously, for non-indentation, we need a flat surface with the C-axis in plane, parallel to the plane of the polishing plane, and perpendicular to the indent. And so that's why we call this Geo 90, because it's geological Aragonite, and 90 degree between the orientation of the indenter or the indentation, which in this case is vertical, and the orientation of the C-axis, which is horizontal. So that's Geo 90. 45 degrees is obviously 45, and 0 degrees is with the C-axis vertical. That's parallel to the direction of indentation. These are the prisms oriented as I just described. Connor's synthetic spherulites instead show this enormously larger apparent fracture measured as I just described with E over H square root. So synthetic spherulites have those tiny little misorientation angles of adjacent crystals, as is intuitive because these are just the radii in a sphere. Obviously, the adjacent ones have this small angle with respect to each other, and the toughness is enormous compared to the geologic one, significantly greater, even though the error bars are large in this case because the spherulites are small. They're on the order of 10 to 50 micron in size. The error bar is large, but the difference is significant. Interestingly, if we look at Aragonite skeletons from corals, we see that they're, of course, all made of spherulites, and they also are significantly tougher in this apparent toughness than geologic Aragonite in any orientation. Then we look at nacre from the Tahitian pearl oyster, and we look at the orientation with the C-axis vertical. So exactly as you look at a shell, as a shark would go and bite on a shell, and that's the direction in which it needs to be tough to protect itself from predators and from the attack of animals. You see significantly greater toughness compared to the single crystalline Aragonite. When we put the shell in the on-edge direction instead, I call this PM for Pinctada margaritifera 0 because of the C-axis orientation, or 90, this is when nacre looks like a brick wall in the embedded and polished sample, we see much lower. toughness comparable to the single crystalline aragonite. Red abalone from California, instead, is a superstar of all of the minerals and biominerals. And in fact, if you look at why it's the most studied and the most famous of all biominerals, abalone maker is the superstar. It is the toughest of all of them, and our experiments confirm that brilliantly. And again, if we analyze it at 90 degrees between C-axis average orientation and indentation, we find much lower toughness. So in case you were disturbed by all of these images, that's a clean graph for you. And clearly the superstars are Connors, synthetic aragonite spherulites, and the 3 superstar biominerals. They have this small misorientation of adjacent tablets. Therefore, the slight misorientation toughening appears to be confirmed. Of course, we still need to measure rigorously the crack length before we can make this 100% quantitative and rigorous, but preliminary data appear to indicate that it's indeed the case. And if true, this really confirms why it evolved convergently. I have 4 more minutes, so I'm gonna skip this and go to the conclusions. What we found is that calcium carbonate biominerals have small misorientations of adjacent crystals, and this toughens the material. It makes it more resistant to fracture, and we verified this with molecular dynamic simulations, and the preliminary data from nanoindentation appear to support that conclusion, that hypothesis. What it means in biology is that the evolutionary advantage and convergence, and importantly, even beyond calcium carbonate, I'm going to show you a few data from teeth that actually also show this small misorientation of adjacent crystals, and that makes this even more relevant, more general to biomineralization. And in material science, there is a greater toughness and from 1 to 30 degree boundaries instead of the classical so-called low angle boundaries that are usually between 1 and 15 degree misorientation. So that's a new way of looking at low angle boundaries. Open questions are, how do organisms do it? Exactly. We don't know how they impose this small misorientation angles to their growing crystals. Can we do it? Can we understand it and reproduce it in vitro? Can we do synthesis of materials that have this small misorientation? Can we do 3D printing of small misorientation materials? If the answer to these questions is yes, it will be super interesting, because obviously biominerals are formed in environmentally friendly conditions. So we could, in principle, do green chemistry, green synthesis in environmentally friendly ways of producing materials instead of doing the sintering and all sorts of other systems that for industrial production that currently use high temperature, high pressure, harsh chemicals, and so on and so forth. So protecting environment is important. I promised to go beyond calcium carbonate, and in the remaining 2 minutes, I'd like to cruise through that and show you that eggshells grow very much spheroidically. They can be made of calcite or aragonite, depending if they are avian eggshells from birds or turtle eggshells. And they start from spherulites, and then they grow columns, and you can see them from the morphology in the scanning electron microscope very clearly. You can see it in EBSD. You can see it in dinosaur eggshells as well. And EBSD shows that adjacent crystals have small misorientations. So this appears to be a bigger, more widely diffused strategy to toughen biominerals. This is from human enamel in our teeth. And you can see these structures called the rods that are actually gradually changing color in a pick map, which means gradually changing orientation. And so you see that within this one rod, there are lots and lots of whisker crystals parallel to one another into these tubes called rods. And they are 50 nanometer wide and many, many microns long. And these rods lie in the plane of the image here at bottom left, at top right of the image, and towards the center of the image. They're perpendicular to the image plane. You see that they vary gradually in color from magenta to blue to cyan. So magenta to blue to cyan, that's a 60-degree angle. And here you go from black to red, that's a 90-degree angle. Here you go from magenta to blue to cyan again, and so on and so forth. You can see very gradually changing colors. Where there's an abrupt change in color, like here, when you go from this very dark blue or cyan to green, this is a bright green to very dark color, that's an almost 90-degree angle right there, and right here, and right here, that's where an organic sheet appears, just like in the mollusk shells it happened. And the rest of the changes are gradual. So in collaboration with Ling Li, we looked at this fantastic fish that's called the black drum, and the black drum fish, and it crushes shells, and it has these teeth that are unbelievably stiff and hard, but mostly stiff. They're the stiffest biomineral thus far measured, and they're optimized to crush shells. This is how the fish feeds itself. And if we look at the structure with a pick map, with pick mapping, we can see that adjacent crystals have narrowly misoriented crystals. And so again, you see magenta to blue, and here you see yellow to green, and all of this are 30 or so degree angles. So this seems to verify what we found in the carbonates, and then in hydroxyapatite. So this case is actually fluorapatite. There's a typo there. This should be fluorapatite. In the case of human enamel, it's hydroxyapatite. So this is well beyond carbonates, but fluorapatite in the fish teeth. This is the same thing for shark teeth, for example, for parrotfish teeth that you see here, and quite a few other animals. So mouse teeth, hydroxyapatite, parrotfish, fluorapatite, sheep is hydroxyapatite, shark teeth are again fluorapatite. So well beyond the carbonates, we always find the same thing. If we look at the distributions of misorientations, they're all narrow, they're all below 30, 35 degrees. So with this, I'd like to thank my group, especially Kayla and Connor, whom I've already mentioned, Cheng Yu, that did much of the work on coral, and others whose work I didn't present, and many fantastic underrepresented minority undergraduate students who are at UW Medicine and bring a lot of energy and enthusiasm to my group. And we have them do massively parallel data processing so that we make sure that all of the decision making that's involved in data processing is done by different people, creative, talented people that and if once we converge towards a solution that multiple people in multiple ways have found, then we embrace that as the most reliable one and find the courage to publish it. I'd like to thank lots of fantastic collaborators, Marcos and Davide, whom I've already mentioned, Tali and Sylvie, who are two fantastic marine biologists, Ilya, who taught me everything I know about human teeth, Mikkel and Admir, with whom I collaborate on Nacre, Ling, with whom I collaborate on drumfish teeth, Laszlo, with whom I collaborate on spherulites, and he's a face field theorist. And we've understood theoretically and experimentally how spherulites nucleate and grow with Laszlo, which has been a fantastic piece of work this year. Susanna Porter knows everything there is to know about microfossils from 500 million years ago. Christine Bergman is a world expert on clump isotope thermometry. Andy Knoll is the best paleontologist in the world. He taught me everything I know about paleontology and the history of life and convergence. And I had the privilege of calling him a collaborator and a friend and a very frequent co-author in publications. So I'm super thrilled to work with Andy. Yael on sea urchin spines, Jim on particle attachment, Kevin on apatite spectroscopy and simulation on the apatite spectra, Boaz on the Vaterite spicules, and Ali on the parrotfish tooth. And I'm going to leave you with a beautiful image that shows that Nacre starts from spherulites and then becomes layered and columnar in California red abalone. And finally, thank you for your attention.
That's really fantastic talk. So I really, draft with you all these wonderful pictures. Congratulations, Pupa. Yeah, we're proud of you.
Thank you.
Yeah, so nice. That's now let's go to the panel discussion. First, let me introduce our panelists. First is Michael, our older friends. Yeah, actually, I'm so proud. This time I want to say thank you, Michael. You introduce Pupa. Pupa is the best speakers. Thank you. Yeah. So Michael actually as older friends, he was last year he jumped in ICANX talks and for the light, the Young Scientist Award, right? Yeah, and then he gave a talk on ICANX talks. He's a professor in University of Medicine, Wisconsin, and it developed many new technologies, and he's very nice speakers. So welcome, Professor Michael Katz. And our next panelist is Yurong Ma. Yurong has her PhD from Peking University in chemistry school, and I spent 10 years in Peking University and working very fantastic jobs. And now he is a professor, a full professor in Beijing Institute of Technology. I stayed in the field very close to Professor Pupa. So welcome, Yurong. And we also welcome our last panelist is Ling Li. I think you already heard his name several times in Pupa's talk. Yeah, wonderful collaborators and many interesting works. And Ling got PhD from MIT and then spent a few years in Harvard as a postdoc. And now is assistant professor in Virginia Tech, is doing a lot of fantastic work. So let's welcome all of them. Paul, please start our panel discussion.
Terrific. Actually, since I will have to evacuate during the discussion, I wanted to ask you something first. That it struck me how ubiquitous calcium carbonate is, and you've spent so much time thinking about it. If there are special properties that it has, and I'm wondering if the organics, if there is some pattern, if you will, to the organics that are used in the connections, and if there's some repetition as well there amongst either materials or functional groups or something that binds to carbonates. And
Beautiful question. How many years do you have before your flight?
Oh yeah, well, I think I seem to remember you have an excellent bourbon, and we can continue the discussion when we get together in person again. But let's get started.
It's true. I forgot you're tasting expensive bourbons. Okay, so so brilliant question, Paul, as usual. Thank you. Importantly, calcium carbonate is cheap, readily available in nature, and it's ubiquitous. And so it's very easy for organisms to use it to make their skeletons and their skeletal parts and their cells. So that's one reason. The other reason from our point of view, not the organisms, but it's part of the carbon cycle. And so it's our great hope that it will actually remove more CO2 from the atmosphere, which is amazing. The other part of your question is the organics and what's repeating among them, are there functional groups and so on. So one interesting thing is that there are lots of negatively charged amino acids in the protein sequences in all of the mollusk shells, but not, and that would seem to indicate that they're great calcium binders because they had these negatively charged amino acids and aspartate and glutamate, and they therefore attract positively charged calcium ions from solution. That seems to be an indication. However, then when you look at the echinoderms, they don't have negatively charged proteins. When you look at cnidarians, they don't either. And so it may be necessary, certainly not sufficient. You see what I mean? It's just not ubiquitous. It's an indication, but it's not by any means general. Other things that are interesting to notice is that no two proteins across any two biominerals are ever in common. Super disappointing. Not even Maker, that looks morphologically the same and it seems to have evolved in such similar ways. Absolutely no two proteins in common across any of the classes, which is super annoying because you can't figure out what the guidelines are, right? So, but that's another indication that they evolved divergently and then they converged towards making Maker. There are no common ancestors and proteins conserved by them.
Wow. Well, thank you. Thank you so much. And I will look forward to continuing that discussion. I do have things to follow up on, but let's move to Professor Ma and ask if, let her begin the discussion.
Okay, Pupa, it's wonderful work. I really wonder, so you mentioned that in the end, the nature guided to form misorientation because of the higher mechanical property, right?
I think so. I don't know. It could be a self-assembly method. It's not necessarily guided. In the case of spherulites, for example, we can make it happen without any guidance. It just happens by itself. So it's not necessarily guided.
I just use the word guide that you used in your presentation because is really conventionally to get this conclusion. But is it also possible to consider that because the nature, the organisms form minerals via amorphous precursor, in the end, they end up with crystals with misorientation because of the energy or fast growth process. Maybe this is kind of the side byproduct of the mineralization process. But anyway, in the end, it has higher mechanical property. What I want to say is the formation process, the amorphous precursor induce or maybe plays a key role to form crystals with misorientation.
I love this question. You are brilliant as usual. You ask the best questions. So yeah, interesting. I've been thinking this for years. How do I put these two stories, which I've studied completely independently, the amorphous precursors and the misorientations. I've studied both of them for years in a variety of different organisms. And I had never found a way to actually put them together, but I've always thought that the misorientation is a consequence, exactly as you say, of formation from an amorphous precursor. There is a paper that came out this year, the paper I mentioned by Igor Zlotnikov in Advanced Materials, that says that when a material crystallizes from an amorphous precursor, it shrinks in volume. And when it shrinks in volume, it forms the so-called misfit dislocations, which, and now it's my interpretation of his work, he doesn't say it, but it would actually shrink in this direction. Say that, for example, I'm the crystal and this is my growth front, and this particle of my front surface is crystallizing, it always shrinks towards me, towards me, towards me, towards me. So this would generate a gradient, right, in all of those dislocations, and those would always be oriented in one particle in a certain direction, and in the next particle in the same direction, and then in the same direction, the same direction. And so part of it is that the crystallinity propagates from one to the other, and part of it is that this misfit dislocations actually pile up, and that's what generates the gradient. So that's my interpretation, is not Igor's, but my interpretation is that it could happen that way. How we could go about demonstrating that is very tricky. So if you can think of a way you're doing a synthetic system to verify that, that would be super awesome. We should work together.
Thank you. I totally, and anyway, I agree that in the end, nature chooses to form minerals with high mechanical property. In the end, they can protect themselves. That's genetic part. Thank you. Wonderful work. Congratulations.
Thank you.
I'm going to excuse myself. My car is here, and I'll turn things over to Alice. Fantastic talk and fantastic discussion. Look forward to much more.
Okay, Paul, take care. Have a safe trip.
Bye.
Thank you.
Okay, so yeah, let's continue. Yuzhong, did you have any further questions?
No.
Okay, so let's move to Michael. Yeah, so your collaborators. Yeah. So,
sure. Thank you so much, Alice, for bringing us together for this great event, as always. And Pupa, thank you for your beautiful presentation. It's always a thrill to learn from you. I have, I guess, a couple of questions. One a little bit more technical, one a little bit broader. Maybe they're both broader, really. The first one is, do you think there are any evolutionary trade-offs to nacre formation? You, I think, pretty clearly convinced us that you can get enhanced toughness with these, and I don't mean nacre formation, maybe evolutionary trade-offs to this misorientation, small angle misorientations that you noticed. So you pretty clearly showed us that you have enhanced toughness, but presumably that's not the only figure of merit for all biominerals. Maybe you get enhanced toughness, but then you lose something else, or something else is reduced. So what do you think? What are the different figures of merit, or do you think there's anything that trades off with this enhanced toughness that you observe?
That's a great question. I don't know how to give you a quantitative answer. One thing that surprised me tremendously in this nano-indentation experiments is that when we measured the hardness and the stiffness, so this would be each H and E, Young's modulus with nano-indentation, both H and E are lower than in the geologic single crystals. I was super disappointed about this. But then when you do the ratio and apply exponents, then they win. And that probably means that there's something organic in there that makes them compliant and so less hard and less stiff. Therefore, that could be the trade-off. So if you can think of a system that's not fracture, but that's something else that will require either hardness or stiffness, then they lose for sure. So I guess that's the most quantitative answer I can give you, I think. Mm-hmm.
Yeah, that's very interesting. Actually, maybe then one more kind of follow-up, I guess, technical question. So, I mean, you've it seems clear that there's again this evolutionary advantage to these misorientations, and you said in your conclusion that how biology selects for these small misorientations is an open question. But yet, at the same time, you also said that you've been able to grow these artificial spherulites in the lab with similar properties. Can you tell us a little bit more about this, about the growth mechanism in your lab? Because maybe that sheds a little bit of light. This is something that you're able to do yourself, right?
Yes, but I don't know that I can actually shed light on the biological mechanism. Let me say why. So the way a spherulite grows is the number one requirement to obtain spherulitic growth in any material, being it aspirin, chocolate. Turns out that spherulites are incredibly common all around us.
You said chocolate?
Yeah, cocoa butter,
Okay.
sugar, sucrose is actually that half fructose and half the other one. Glucose they're both spherulitic crystals. It's unbelievable how ubiquitous spherulites are. This is why we spent so much time with the phase field theory and experiments to understand nucleation and growth of crystals of spherulite. So the number one important requirement, and probably the only requirement, is that a crystal needs to have a propensity for growing acicularly. So it wants to grow long with a long aspect ratio, make a needle. If it wants to make a needle, and Aragonite wants to make needles, then it will grow spherulitically. It can nucleate on a co-oriented or misoriented crystal, doesn't matter. The crystal orientations that win are the ones that grow radially, simply because they have space to grow in the radial direction. If they're slightly misoriented, they bump into each other and they don't grow big. This is surprising. We looked at spherulites at very high resolution and nanoscale with pick mapping with down to 10 nanometer pixels, and we could not find what I expected, which was randomly oriented crystals that would run into each other, stay small, therefore not become the dominant component of the space filling of the spherulite, but still be there. They're not there. That's the crazy thing. In spherulites, whether you look at chocolate, aspirin, if you look at the majority of coral skeletons, they're not there. What actually made us figure out exactly how it happens in spherulites for them to grow is the completely fortuitous discovery that in a few species of corals, for reasons we don't really know, some of the nuclei that are misoriented stay. Probably they are kinetically trapped. There's some organic that keeps them there, and so they don't get coarsened, because that's the thing that happens during spherulitic growth. Not only acicular, but also coarsening. So coarsening means that bigger crystals grow bigger at the expense of the smaller ones. So in a system of nanoparticles, for example, when you have coarsening, the grain sizes gets bigger. And when you have smaller other ones, those disappear, they dissolve, and the big one grows bigger. When you're talking about the spherulite, if you had these randomly oriented crystals, which I call sprinkles, because in a pick map, they look like they have random orientations, and therefore they look like sprinkles on a cupcake or on a cake, right? Those tiny little sugar little things that have lots and lots of different colors. Okay, so these sprinkles are the nucleation events or testimonials or the crystals that remain from the original nucleation events. They do not get coarsened. But this is actually how all of the spherulites grow, whether they are chocolate, sugar, aspirin, or any other drug. They all nucleate with random orientations. but then the ones that are not oriented radially bump into each other, don't grow big, remain small. The other ones grow acicularly, they are oriented radially, they have space to grow, they grow bigger. And so in the coarsening process, they win, and the small ones disappear. So that's what I know about spherulitic growth. How it actually happens in coral skeleton is pretty clear. It's exactly this way. How it happens in nacre, or how it happens in teeth, is a little bit different than that. So, for example, this acicular growth that we can see in the spherulites at the beginning of nacre, in the image I concluded with, these are definitely acicular crystals. But then we demonstrated that in the layered nacre portion, the crystals can only grow the way they do and generate the patterns of crystal orientations that we observe if they grow slow along the C-axis and fast along A and B. And that's pretty clear. If you want to fill a space that's short vertically and very wide horizontally in both directions, you want to grow fast along A and B and slow along C, because it's how you can space fill faster. And that's totally counterintuitive. What's this switch between growing faster along the C-axis? And Aragonite does 10 times faster along the C-axis than along A and B. And then in nacre is exactly the opposite. And how does that switch occur? There's got to be a protein that slows down the growth along the C-axis direction that does not slow it down along A and B.
Okay, really great.
Yeah. That's fascinating. A lot of mysteries.
Yeah, exactly.
I will keep my broad question for some other day. Save some time
feel free to ask it. I'm
okay.
happy.
Yeah, so we can move to Ling. Yeah, so Ling, please.
Sure. Yeah, thank you, Pupa. Always fascinating and inspiring. It's always nice to hear your talk. So because of time, I'll just start with a short question. So what do you think in terms of the material in between the adjacent grains where you have the misorientations? Is it there are some organics or amorphous materials, or just inorganic mineral directly in contact with adjacent grain? So what is the structure at the interface?
That's a brilliant question. I can't answer it because I don't have enough resolution with thick mapping. If you're an expert with TEM, we could actually look at the dislocations, because I've been wondering exactly the same question. And it's clearly a 3D TEM system to study. And so one needs to do tomography and diffraction and have atomic resolution. So it's a slightly different experiment. It takes difficult experiment, and it takes a real expert at TEM tomography and crystallography at the same time. So it's a little bit of a niche. There are very few people I know that know how to do this. Do you?
Yeah, I know it's difficult, but I guess.
But to answer your question, I don't think there are organics between these slightly misoriented crystals. And the reason for that, and that's why I wanted to study dislocations to understand, because they obviously must be growing from different sides and converge to one another there, or one imparts its crystal orientation to the next one, but slightly off, probably because of dislocation or because fluxes of materials, chemotaxis, something. The reason I think there are no organics is that there are other places where there are organics, and that's where we observe the big misorientations. And this is true in nacre, it's true in teeth, it's true in the prismatic layer of the mollusk shells, and blah, blah, blah. You name it, you will find it. In corals, very much so. And so at the boundary, so those things called the trabeculae, each bundle of crystals that grow acicularly, then it ends with an organic layer, and that's where the angles are big, the variation of from one spherolytic bundle to another. Between two spherolytic bundles, there are no organics. And at least none that we can see with 10 nanometer resolution with thick mapping or 20 nanometer resolution with thick mapping. And we can't rule out that there are single molecule ones, but at the very least, they're not the leading mechanism for misorientation. So that answers your question.
Yeah, great. Thank you, Pupa. Do I have time for another one?
Okay, so I may I say something?
Do we have to stop at 8:30?
Okay.
I can reach
so
out to
because
you.
Yurong have some urgent, will leave to another meeting and something. So almost time. So Ling, you can send your question to Pupa. Okay, sure. Later. Yeah. Okay. Thank you very much for this. Wonderful panel discussion. I feel so great, Pupa, your talk. It's amazing. I see so many beautiful things, the arts, everything. Yeah, thank you all of you. Yeah, I'm looking forward to have all of you talk on this, I can ask talks. So Ling and Yurong must be prepared. Okay, thank you. Yeah.
Yeah, thank you, Alice. Thank you, Pupa. It's nice to see you.
Yeah.
Great to see. It was fantastic privilege.
Don't worry, don't move, Pupa. I still have my certification to you. Okay. Yeah, Pupa, I feel so great. Yeah, as I can ask, I will deliver this certification to you. Yeah, wonderful talk. Pupa, we're proud of that. All these, fantastic technologies. We're looking forward to, see them in the future. Yeah, more good things and moving forward to get the team grow up. Yeah, more and more people jump in that. Like you say in the very beginning, maybe not a lot of people know this, but now I think many people love it. Yeah.
All right. I am delighted.
Okay. Yeah. So I will deliver this. Yeah, the e-version to you next time when I met you. I will, give you the hard copy. Yeah. Thank you. Perfect. I also want partly to give a hug to you. Yeah.
Perfect.
So, okay. Thank you very much. Yeah.
Thank you very much.
Very wonderful panel discussion. And so next year we're going to have Cheryl Gomez on this stage, and she's going to talk a fantastic talk about assembly engineering for the particles. Yeah. So I think this week we have for about the crystal. Yeah. Next year we will to hear another story from this wonderful lady scientist, the female scientist Sharon from Michigan. So yeah, let's see everyone next week. And we have our Young Scientist Award. You see that 21 young scientists was chosen as a finalist of this year. So yeah, the week after we will have all of them to get on the stage to do the final, presentation, and we see we got top 10 to be awarded. So we are looking forward to have all of them. We're looking forward to have the talks. Okay, that's the end of today. Thank you very much. I see you next week, and I see all of you soon.